Namaskar, welcome to PK's classes. Today we'll study about the mechanism of fluoroquinolones. We'll study also the classification of quinolones and fluoroquinolones. So the first generation of these drugs are the quinolones, for example nalidic acid, pipemidic acid. Then the second generation ciprofloxacin, ofloxacin, norfloxacin, lomefloxacin. Third generation levofloxacin, gotifloxacin, sparfloxacin. This moxi and gemifloxacin are also third generation. This trovafloxacin is basically the fourth generation one. And sinoxacin is another first generation one. And generation wise the spectrum increases. And this is the general structure of the fluoroquinolone, and different derivatives of this fluoroquinolone scaffold leads to all the structures like nalidic acid, ofloxacin, ciprofloxacin, levofloxacin, moxifloxacin, sitafloxacin. So fluoroquinolones show their action by binding to two enzymes. One is topoisomerase 2, also known as DNA gyrase, and topoisomerase 4. And mostly in gram-negative organisms, topoisomerase 2 or DNA gyrase. In gram-positive organisms, topoisomerase 4. So this causes the broken DNA strands. There will be no DNA replication, so cell death occurs. So this is the mechanism of fluoroquinolones. So as far as this topoisomerase is concerned, there are 4 isomers of topoisomerase, and these topoisomerase enzymes control and modify the topological states of the DNA in cells. Topoisomerase 1 and 3 catalyze merely the relaxation of DNA, whereas topoisomerase 2 catalyze the supercoiling of DNA, and topoisomerase 4 is involved in separation process of the daughter chains after chromosome duplication. And in bacteria, these two enzymes, topoisomerase 2 and 4, DNA gyrase and topoisomerase 4, are present, and quinolone and fluoroquinolones, they inhibit these two enzymes. DNA gyrase in gram-negative, topoisomerase 4 in gram-positive. By inhibition of DNA gyrase, they prevent the relaxation of the supercoiled DNA, and this relaxed DNA is required for the normal transcription and replication. And by inhibition of topoisomerase 4, it interferes with the separation of replicated chromosomal DNA into respective daughter cells during cell division. So let us discuss these two enzymes one by one. And in bacteria we have this circular DNA, and this circular DNA, when it is coiled one, so this is DNA structure, this is coiled one, and supercoiled, as you know, the chromosome is compact because of the supercoiling. And let us see what is this supercoiling about. So this is your relaxed DNA, and this is supercoiled DNA, and you can see this in a telephone cord. So this is relaxed one, and this is the supercoiled one. Okay. So the relaxed circular DNA, when supercoiled, this looks like this. And if you give it a cut, this will be the linear DNA. So this supercoiled DNA, that means this circular DNA is coiled. So this circular DNA is coiled, that causes the supercoiled DNA. And this supercoiled DNA is converted to the circular DNA, the relaxed DNA, by means of DNA gyrase. And during the replication, the two circular DNA are interlinked, catenated, and to separate these two we need topoisomerase 4. Even if it is knotted, that is also we need topoisomerase 4. So this is the relaxed DNA, this is the relaxed DNA, and this is the supercoiled DNA. And DNA gyrase helps in conversion of the supercoiled DNA into relaxed DNA. And DNA gyrase has 4 genes, 2 gyrase A genes and 2 gyrase B genes, and this is the structure of the DNA gyrase. And that DNA gyrase separates the supercoiled DNA so that there will be relaxed DNA, then only there can be DNA replication. Suppose this is one DNA and this is intercoiled with another DNA. So that separation, this is supercoiled, that separation will be done by this DNA gyrase. Then this will be separated like this. So let us see the mechanism of action. So suppose this is the supercoiled DNA, the G segment and T segment, and this is ATP dependent DNA gyrase or topoisomerase 2. So this supercoiling will be separated and we will have the two separated coils. So this is another structure, another figure where the supercoiled DNA will be separated into two different DNAs. So DNA gyrase converts this supercoiled DNA into relaxed DNA so that this relaxed DNA can undergo replication, and during the replication we can get two daughter DNAs which are catenated, and that catenated DNA will be converted to two separate daughter DNA or decatenated DNA by topoisomerase 4. This topoisomerase 4 is encoded by 4 genes, 2 par C and 2 par E genes. So topoisomerase 4 has 4 parts, 2 par C and 2 par E, and so 2 genes are there, par C and par E. And you can see this is the relaxed one. This is a supercoiled DNA, decatenated relaxed DNA, and catenated DNA after replication. So when from one DNA we have two DNA because of the replication, these two DNA may be catenated and we need to separate, and that separation is done by topoisomerase 4. Suppose this is the topoisomerase 4 enzyme, and topoisomerase 4 will bind to one of the daughter DNA, and then you can see it will go like this, and then these two will be separated and the enzyme is also separated. So it will first bind to one of them, then it will become the interlink between the two, then it will separate out and we will have the two daughter DNA. And this is how topoisomerase 4 acts. So as we have discussed, the DNA gyrase prevent relaxation of supercoiled DNA that is required for the normal transcription and replication. And the inhibition of topoisomerase 4 interferes with the separation of replicated chromosome and DNA into respective daughter cells. So this is all about the mechanism of action. This is the action of DNA gyrase, the topoisomerase 2, and this is the action of topoisomerase 4. Thank you.